Okay, so welcome everyone. I'm excited to be back here for an Eric and Wendy Schmidt Center joined with MIT EECS Colloquium, where we feature speakers who either think about foundations of machine learning and bring it into important biomedical questions, and also go the other way around of thinking about important biomedical questions and then develop new machine learning algorithms tailored for these particular questions. And so I'm very excited to have today Emily Fox here. So she is a professor in the Department of Statistics and in the Department of Computer Science at Stanford, and then also Chief Technical Advisor at insitro. She knows this area very well, did her undergrad and PhD at MIT, was then also professor at the University of Washington before moving to Stanford, also led the health AI team at Apple while at the University of Washington as well. Has received many, many prizes from Sloan Research Award, NSF Career Award, etc., etc. But I'm very excited to have her here. She thinks very deeply about new methods in machine learning statistics, particularly where you have a lot of sequence information, so time information, and then also thinking about uncertainty, so using Bayesian approaches, developing new Bayesian approaches, and then also generative models to really understand complex or informative latent structure that is behind complex, high-dimensional, multimodal data, be it from genomics to neuroscience to health data, like what you would get from wearables. So I'm very excited about this talk, where I'm sure we'll be going through the whole arc, and in particular then getting at causal information in the end of thinking about how to intervene in the system. So thank you, Emily.
Well, thanks, Caroline. Very excited to be here. Definitely a trip down memory lane for me, being so close to MIT. And so in this talk, I'm going to discuss both ongoing work at insitro as well as really just a vignette from my lab at Stanford. And both parts of the talk really focus on this theme of how machine learning can be transformative for biohealth, from drug discovery to health monitoring. But looking and focusing just on predictive performance really is not sufficient. And getting to causality is key to get to decision-ready science. Okay, to start, let's discuss some of our ongoing work at insitro on disentangling causality in drug discovery. And the first thing that's important to note is that drug discovery is slow and expensive, and it's only getting slower and more expensive. And the vast majority of drugs end up failing in the clinic, with roughly only about 10% getting to that final approval stage. And so the question is, what's going on here? Well, there are really three key stages to delivering a drug, starting from discovering the therapeutic target to developing the molecular matter that acts upon it, to eventually taking that drug to the clinic in human clinical trials. And there are many root causes of that eventual failure that appear at each of these different stages. And although insitro works on all stages of this pipeline, I'm going to focus in this talk on the target discovery phase. And the summary of the root causes here really boils down to there being a weak causal linkage between the discovered target and the clinical outcome of interest. Okay, so to better understand this, first we need to understand the ways in which target discovery is done. And there's a whole range of different methods, going from looking at simple cellular systems to more complex animal models, to looking at human disease cohort data. And as we go across this spectrum, there's a trade-off in terms of the level of translation to human biology versus the level of mechanistic control. And unfortunately, when we go to the human disease cohort end of the spectrum, that source, which is obviously closest to human biology, is also most susceptible to various sources of confounding and bias. Nevertheless, multiple meta-analyses have shown that targets that have human genetic support have 2 to 3 times the probability of success in the clinic. So at insitro, we're really focused on leveraging AI to amplify the power of human genetics to get at these novel therapeutic hypotheses with even higher probability of success. Okay, well, of course, everybody is talking about the AI revolution, but what's critical here is the confluence with the biology revolution. So to get at these novel therapeutic hypotheses, we're really leveraging the wealth of different clinical modalities that are increasingly being collected together with human genetics. And the human genetics represent our experiments of nature. And then we're using that to try and get at causal genetic drivers of disease. We then build confidence in these targets as well as explore other potential targets, leveraging perturbation screens in the lab, where we perturb a set of cells in some disease-relevant cell model and look at the phenotypic consequences using a range of modalities ranging from high-resolution microscopy to single-cell and bulk omics. And leveraging machine learning to extract and amplify this disease-relevant signal to get to these novel therapeutic hypotheses really does show a lot of promise. But as I said, it's really not sufficient in and of itself, because getting to causality is key to our probability of success. And even with experimentation, experiments of nature, our controlled experiments in the lab, trying to suss out causality is challenging. Biology is messy. So really what I'm going to do here is take you on a tour of some of our science and some of the approaches that we've developed to try and tackle these challenges. Okay, so let's start by discussing our approaches for target discovery, leveraging clinical data with human genetics. And a typical approach here is to start with a genome-wide association study where you define some phenotypic trait relevant to the clinical outcome of interest and look at locations across the chromosome for significant associations with that phenotypic trait. And we all know association is not equal to causation, but DNA is upstream of disease. So are we okay to call these GWAS hits causal variants? They're often referred to as such. And to think about this a little bit more deeply, let's look at an intuitive example of trying to find genetic drivers of obesity. And what seems like a straightforward association study presents a number of challenges. One is through confounding, where, as an example, proximal genes can have correlated haplotypes, but their causal function can be completely distinct. Another challenge is through mediation, where our GWAS is really looking at lifelong effects. So some gene may be causal of childhood obesity, which may lead to adult obesity, but modifying that gene directly may have no direct effect on adult obesity. Okay, another challenge is the fact that our diseases are highly multifactorial. So for example, in obesity, there are lots of heritable factors that influence things like how much we like to exercise or the foods that we like to eat that also influence obesity beyond the metabolic path of interest. And even if you magically had the data available to control for all these factors, very likely there's going to be some unknown confounder that opens up this backdoor path leading to collider bias. So as an example of this, it may seem very reasonable when studying obesity to want to control for the effects of exercise. So we run a study, we give everybody Apple Watches, we condition on this data, and in that data is resting heart rate. And resting heart rate is not causal of obesity, but stress is causal of resting heart rate and is also causal of obesity. So this gene, G prime here, looks like a promising target. But if you're about to go spend tens of millions of dollars taking this to the clinic, how do you feel about G versus G prime? Okay, well, up to this point, I've yet to clearly define the phenotypic trait that we're using in our GWAS. And as I'll discuss, the choices that we make here can have very important consequences in terms of the level and sources of bias that we're faced with. Okay, well, in many scenarios, we actually use what's called a proxy phenotype in place of some clinical gold standard trait that may be very challenging to directly measure. So in obesity, a common proxy phenotype is looking at BMI, but this is a very crude measure, just being weight divided by height squared. So this bodybuilder can have the same BMI as this adipose person. Likewise, BMI is susceptible to all the sources of bias that I just discussed because there are many heritable factors that also influence BMI. So instead, what we really want to define is a phenotypic trait or a proxy phenotype that has more specificity to the causal path of interest. So as an alternative to BMI, one can consider waist to hip ratio. This is still a rudimentary measure, but it better approximates abdominal fat that's associated with the kinds of negative metabolic and cardiovascular outcomes we're interested in. However, as we showed in this paper, using ratio traits can be problematic because the discovered associations can be due to causal relationships just with the denominator. And typically, when we're using these kind of ratio traits, we're interested in the numerator adjusted for the denominator. And also, as discussed in this paper, directly taking that approach tends to be preferred. But an alternative approach is to leverage the rich clinical imaging modalities that we have available to extract phenotypic traits that are better aligned to our biological hypotheses. So for example, from MRI, we can derive fat volume in specific depots or fat fraction. And our ClinML platform enables custom annotation and active learning cycles to train segmentation models that allow us to rapidly derive these types of image-derived phenotypes with strong disease relevance. Okay, well, beyond the precision of our phenotype, we also really care about its scale in order to be able to power our GWAS. And one challenge of using these image-derived phenotypes is that typically we only have relatively small or moderate sample sizes available of that imaging modality. And so we end up with this precise phenotype, but one that's underpowered for our association study. So, we may only uncover the targets with the strongest associations, and those have been analyzed many times over. So instead, what we do is we train machine learning models to predict our precision phenotype from much more commonly available data modalities. So an example of this that we did that I can talk about here is in our study of MASH, or fatty liver disease, where the clinical gold standard trait is MRI-derived PDFF, and we train models to predict PDFF from DEXA, a bone density scan that's more widely available, or blood biomarkers and metabolites. And what we see is that if we run our GWAS on the clinical gold standard PDFF, we get some significant associations. But as we move to imputed PDFF from these more widely available data modalities, we see this dramatic increase in the number of significant loci. And these aren't just any loci. A number of these have actually been credentialed using other approaches. But this represented the first time of having human genetic support behind them. And FGF21 is an example of that. And we also discovered a number of novel targets that we've since passed to in vitro and in vivo credentialing. But before I discuss that, I just want to mention that there are issues with using these imputed ML phenotypes. And one is the fact that it can inflate false discovery rates. And so one thing we do to try and mediate that effect is trying to get to what the causal relationships are underlying those associations. And so for this, we turn to Mendelian randomization, which leverages the fact that our genotype is randomly inherited from our parents. So if our genotype is associated with some biomarker of interest, we can use it as an instrumental variable to help determine whether that biomarker is causal of disease. So one place that we use Mendelian randomization is thinking about reprioritizing our target lists, where we do a bidirectional MR, and we say for each one of the biomarkers that was used to impute liver fat in this case, try to understand whether that biomarker is upstream or downstream of liver fat. Because if a candidate target that we're interested in is associated with some biomarker that we've learned to be downstream of liver fat, we're going to deprioritize that target. Okay, Well, after doing this and a bunch of other post-GWAS analyses, we take our remaining promising candidate targets and pass them to in vitro validation. We use both arrayed and pooled screens. I'll talk more about these capabilities in a little bit. But what we're showing here on the arrayed side is looking at primary hepatocytes under free fatty acid and de novo lipogenesis conditions. And we're taking one of our promising targets, referred to here as MASH1, and doing a MASH1 knockout. And what you see is reduced lipid droplets accumulation uniformly across these conditions. So those are the green dots in the pictures. And then in our pooled screen, we do both a knockout as well as an upregulation of MASH1. And in our upregulation, we see an increase in lipid droplets. So this provides in vitro validation of that MASH1 target, but that's just one target. We did the same type of analysis for a number of targets. And for 4 of our promising targets, we passed it to in vivo validation, in particular looking at a DIO mice, so that's diet-induced obesity. And what we see is that for 3 out of 4 of these targets, we see this reversion of accumulation of triglycerides in the liver. So nice validation of our candidate targets here. Okay, well, let's now turn to the cellular side of the story and talk about our controlled experiments in the lab. And here we're going to ground the discussion in another one of our pipeline projects, which is ALS. And ALS is a deadly disease that progressively kills motor neurons. And the work that I'm going to talk about here is in collaboration with Bristol Myers Squibb. Okay, well, a key pathology of ALS is the mislocalization of this protein TDP-43 from the nucleus to the cytoplasm. And TDP-43 is really critical in a number of RNA splicing activities for critical neuronal genes. And so in its absence, it can lead to the insertion of these cryptic exons that lead to these truncated RNA sequences. So an example is stamin-2, which is a key regulator of axonal health. So for our cellular disease model, what we do is we deplete TDP-43 in iPSC-derived motor neurons, and then we run a whole bunch of perturbations and look at the phenotypic consequences in terms of these stamin-2 cryptic exon and full-length measurements, and see if any of these perturbations revert our sick cells to look healthy again. So a standard screen uses an arrayed platform where within every well we introduce a unique perturbation. And then what we're going to do to create our sick cells is use an siRNA to deplete TDP-43 and then image using high content imaging. But we also want to be able to make sure we're looking specifically at the effects of TDP-43 depletion. So we also introduce healthy controls using a non-targeting SI. And so with these healthy controls in hand, it seems straightforward to estimate our perturbation effects, right? Most of you probably know the answer is no to this question, where the story's really not that simple. There can be very significant well effects where wells close to the edge of the plate have reduced cell viability, confounding our estimates of our stamin-2 measurements. Likewise, there can be significant technical artifacts that lead to really significant plate-to-plate variability, further obscuring these true biological differences. So to ameliorate these issues, an increasingly popular approach is to turn to pooled screens, where we take an entire set of genetic perturbations, introduce them into the same well, and that allows us to look across perturbations all under the same condition. And so then we optically phenotype the cells using core cell morphology, localization of key proteins, and even live cell imaging. And one key thing about the pooled screen is that each perturbation is tagged with a little DNA barcode that we can read off so that we can identify which perturbation each cell had. So we can extract this cell by feature matrix where we have the ID for the perturbation associated with each cell. And machine learning is really critical to this cell featurization process, everything from segmenting individual cells to the barcode calling process itself. And then we can form, like I said, the cell by feature matrix. And in our ALS example, something that would be contained in here are this Statmin2 cryptic exon and full length counts. And what I'm showing in this picture are healthy controls compared to, this is just an example perturbation. And again, remember, we're trying to see if any of these perturbations make our sick cells look healthy again. Okay, so now with this pooled screen, can we estimate our perturbation effects? Well, unfortunately, the saga continues, because just the introduction of the CRISPR machinery can have effects on our cells, such as on cell size or cell viability, which affects cell density, and all these effects can further confound the estimates that we're interested in. So although pooled screens do help with batch effects, it's still important to design appropriate controls. And for this, to account for cell size and cell viability, we stain for GAPDH, which is a housekeeping gene, appears basically everywhere. And so when we're looking at our Statmin2 counts, it's relative to GAPDH. We also introduce intergenic guides, which introduce the CRISPR machinery, but between coding regions, which allows us to account for non-specific effects of CRISPR. Okay, so now with these appropriate controls in hand, we can start to estimate our perturbation effects and provide this ranked list of potential targets to look at. But as we see, a number of targets have just borderline significance. So a question is there any way to extract more signal here? And one important thing to note is that CRISPR has imperfect effects. Some cells may have no effect at all, and some cells may only have a partial effect. So we can really think of CRISPR as an intent to treat. And in our analyses, when we're averaging over cells that may or may not have been perturbed, we're taking a weak signal and diluting it even further. So to address this, we've developed this generative model that says with some probability, a non-control cell may actually not be perturbed. So in particular, we take a contrastive variational autoencoding perspective, trying to disentangle background from salient signal using our control cells. And then for that salient latent space, we model it using a mixture of perturbation-specific Gaussians, and then there's also this default Gaussian that's capturing the effect of all the unperturbed cells. Okay, so we learn the model parameters and infer which cells were perturbed versus unperturbed using variational inference. And we'll start by looking at results on the Replogle publicly available omics dataset. And here what we're showing is just a standard omics preprocessing pipeline where we take our RNA counts, normalize them, log transform, PCA, and then UMAP. And what you can see in the embedding space is completely dominated by cell cycle. It's very hard to separate any meaningful genetic pathway information. But in contrast, when you apply perturb VI to this data, you see that it's really removed that cell cycle effect, and you can see much clearer, I guess the colors aren't super crisp here, but much clearer separation of the genetic pathways. And what I'm showing on the far right is a picture showing the posterior probability of whether the cell was perturbed, and that mass of dark gray dots are all the cells that were inferred as not perturbed. Okay, so here's another visualization of these results where every colored circle is the mean over all cells that receive the same genetic knockdown. And what we see are highly interpretable results, including the fact that this mass of cells that were inferred as not perturbed are all centered around this non-targeting mean here. So we apply perturb VI as a precursor to our estimation of treatment effects. And returning to our ALS example, where again, remember we're interested in this cryptic exon score and full length score, what we see is that perturb VI increases statistical power and assay sensitivity, leading to better estimation of these perturbation effects. And this is results just for an example gene, but we go and do this for all of our genes and show the results here. And when we look at these results, we see this really promising target, AGO2. So are we about to spend $50 million and take this to the clinic? Well, before doing so, despite everything that we've done to this stage, trying to dig in and understand mechanism is still really important. And of course, a full mechanistic understanding is intractable, but there are various things we can do to get to partial mechanistic understanding, including just looking at the literature. And if you look at the literature for AGO2, you will know that it's part of the RISC complex. And what that means is that it actually silences the siRNA's ability to deplete TDP43. That means our ability to create 6 cells in the first place. So no wonder it looks so good, right? But clearly, if you took this to the clinic, it would totally fail. Okay, so filtering out AGO2, I guess I should also mention that we're developing various automated procedures to help with these types of deep dives. But that allows us to filter out targets like AGO2. And for one of our remaining promising targets, which we'll refer to here as ALS1, we've shown that it leads to functional rescue through neuronal regrowth, which is super, super cool. So here we're seeing that the neurite length is statistically indistinguishable from our healthy controls. So this is something we're really excited about. Okay, but so far, when we've talked about the effects of these perturbations, we've talked about them in terms of what we refer to as an anchor phenotype, which is some known biology of the disease, such as stamen2 full-length encryptic exon. But of course, we're also interested in learning novel aspects of disease based on this rich imaging and omics data that we're collecting. So to this end, for both our imaging and omics data sets, we can use self-supervised learning to train embedding models to try and get at machine-learned phenotypes and screen on those. So for example, for our imaging data, we can use a vision transformer as an example. And qualitatively, when we look at the embedding space, so here we're looking at cosine similarity of gene embeddings. And what we see is this embedding space captures known biological processes across multiple different biologies. And importantly, also uncovers insights that were only discovered in the last few years. And in the context of ALS, when we look at our Dino-VIT embeddings, what we see is that from morphological changes alone that are captured in those embeddings, we can predict TDP43 mislocalization or the expression of ALS-relevant genes. And so what this means is that there's something disease-relevant in this embedding space. And so we can take this embedding space and we can learn an axis that takes us from healthy to sick, and think of this as our ML phenotype. And we can look at exemplars on the extremes of this axis and qualitatively describe differences such as soma elongation or reduced neurite density. We can also use conditional generative models to visualize walks along this axis. So it seems promising, but these embeddings are really dense and very hard to interpret in this way because it's really representing a combination of different image features. So to this end, we've been turning to sparse autoencoders, or SAEs, to learn disentangled representations of our embeddings that get to more monosemantic concepts encoded in the embedding space. And we can then look at differentially activated SAE features between healthy and sick, or perturbed and control. And for each one of those features, look at example images of cells that do use that feature or don't use that feature. We can also do conditional image generation on any one of these SAE features. And here for this conditional image generation in our ALS example, this is a visualization of some of our differentially expressed SAE features. We see things corresponding to cryptic exon, localization of key disease-relevant proteins, cell size, and we definitely see much more monosemanticity than we do in the dense embedding space. And so this type of representation is much easier for domain experts to interact with. Okay, we can also use SAEs with our clinical imaging modalities. So we can take SAE features and run GWAS on those latent traits. And this type of approach has really shown promising results in our study of dry AMD using OCT images. But so far, our modeling work has really assumed that we're either looking at our imaging data or our omics data. But of course, we're interested in capturing shared structure that can help disentangle biological signal from technical confounders that are unlikely to appear on both sides. But a challenge with defining these types of multimodal models is, of course, the fact that we have unpaired data. Likewise, morphology and gene expression have just kind of partially shared overlap in the shared biology. And so we want to develop models that are robust to the fact that a perturbation may affect changes in just one modality and not the other. Okay, so our Petri early fusion method for learning these multimodal single-cell embeddings takes inspiration from vision language models that operate over mixed modal documents where the common topic of that document increases the chances of finding that cross-modal alignment. And so Petri conceives of the perturbation label as the topic of the document and pulls cells from both modalities that share a common perturbation. And so the cell embeddings are fused in a cell document using mass joint reconstruction and cross-modal attention. But one of the challenges of using this type of approach is the fact that the sequence length can very rapidly explode well beyond the capacity of standard transformers. And that's because a single cell can be represented by hundreds of image patches or thousands of gene tokens. So to address this, what we do is very aggressive token resampling. So we consider these modality-specific encodings of our imaging and omics data, and then we distill each to a small set of latent tokens. So on the imaging side, we're using a vision transformer, omics side, the perceiver network, and then we use multimodal set transformer to provide critical cross-modal attention. And we're visualizing a couple of these attention heads for a real data analysis that I'll describe in a moment. But you can see the significant cross-modal attention in these off-diagonal blocks. And then using Petri, we can form perturbation level profiles by integrating across cells that have a shared perturbation, regardless of which modality that cell comes from. And we're going to examine Petri and a matched POSH perturb-seq HepG2 dataset, where our imaging data comes from fluorescence and label-free imaging, and our transcriptomics from flex sequencing. Okay, so to start, in order to show some of the benefits of this cross-modal embedding, what we're doing here is we're comparing performance to unimodal baselines, and we're looking at detection of StringDB edges. And what we see is that the Petri model has increased detection or performance of these edges at all false positive rates. So another experiment we ran was we said, okay, let's try and reconstruct masked Bodepi patches, so image patches, so Bodepi stains for lipid droplets. And we're looking at a Petri model that uses or does not use the omics tokens in the model. And what we see here is we're going to start with really an ablation of our Petri model where we train it having randomly permuted assignments of perturbations to cells. So when we're forming these cell documents, there's no meaningful cross-modal alignment. And what we see is that the Petri model using these incorrect labels performs no worse than not having used omics at all. And so this is a positive result and says that these methods are robust to cases where you have disjoint signals between the two modalities. But when you use the correctly trained Petri model with the correct perturbation labels, you see the benefits of using the omics information in imputing image patches. So that's that cross-modal attention coming to play. And we can dig into kind of the biologically meaningful behind this cross-modal attention, looking at the following results, where we're looking at this mass bodopy intensity, and if we use omics tokens for a control perturbation known to increase lipid droplet accumulation, we see an increase in intensity predicted for bodopy. But if we use omics for a control perturbation known to decrease lipids, we see decreased bodopy prediction. We can also use our sparse autoencoders to interpret these multimodal Petri embeddings. And what we're doing here is we're looking, again, at differentially activated SAE features. And for each one of these features, we're looking at the top 3 gene ontology terms enriched in that feature. And we see that we have features corresponding to DNA replication, cholesterol homeostasis, and aerobic respiration, which are relevant in the experimental setup here. And if you look closely at the imaging data, you'll see features that correspond to those descriptors. And the last note is just that these features are indeed capturing multimodal information, as evidenced by the fact that these SAE features are being used both in the omics and the imaging modalities. And this is in contrast to that ablated model that was trained on incorrect perturbation labels. Okay, well, stepping back, what we've discussed is our approaches for leveraging clinical data and cellular data in order to do target discovery. And on each side, we've talked about various sources of bias that arise and some of the challenges at getting to causal therapeutic targets. And we've really just described various ways in which we've chipped away at the block to try to get there. One big lever that we pull is assuming that the sources of bias between these two sides are going to be different. So for targets that have support both on our clinical side and cellular side, we end up with increased confidence on those targets. So for our targets with that human genetic support and orthogonal support from our cellular screens, we then pass them to in vivo credentialing, like I talked about in MASH. And we take the results from this entire pipeline, combine it with evidence, external sources of evidence, and then nominate targets for drug discovery and eventually for clinical development. And this is all in support of in sitro's mission of bringing better drugs faster to the patients who can benefit most using machine learning and data at scale. And of course, I want to thank the in sitro team that's behind all this work. And the talk is not over, so don't get excited yet. There's more. So at this point, I want to turn to some of my group's work at Stanford. And as Caroline mentioned, one of our emphases is on how wearable devices can really transform traditional clinical care models. And one area that we've invested quite a bit in is in the management of diabetes. So the standard of care in diabetes assumes that you have quarterly visits with a doctor, and the goal is to maintain mean glucose within this target band. But of course, between these visits, there can be deterioration in control that goes undetected. But now there are many opportunities for remote sensing based on advances in wearable devices. So for example, glucose can be measured regularly throughout the day, leveraging a continuous glucose monitor, insulin delivered using a smart pump, or activity with a wearable device. And so with this technology, clinicians can now monitor patients remotely from the office and have more potentially informed interventions to give to that patient based on this more frequent and nuanced data that they have available. And this is without the patient even having to come in. Okay, so we all know how we're gonna extract these meaningful insights from this wearable data. We're gonna pass this data to some state-of-the-art black box sequence model, and it's gonna hand the clinician these meaningful insights and personalized recommendations of how to intervene with this patient, right? Okay, hopefully you know that's not where this ends. Okay, so black box models do indeed have good predictive performance, But of course, that does not guarantee that they have good counterfactual simulation capabilities. So to ground the discussion here, let's look at a clinically important case of trying to encourage individuals to exercise with type 1 diabetes to exercise more safely. So activity can increase the risk of hypoglycemia both during and after the period of exercise. And as a result, many patients with type 1 diabetes avoid exercise altogether. So we want models that have good predictive performance. So in this little cartoon example here, this person experiences a hypoglycemic event following exercise, and we like to be able to predict that is going to happen. But to guide decision making, we also want our models to be able to form reliable counterfactual simulations so we can explore other potential interventions, such as reducing insulin at the meal prior to exercise. And maybe this would avoid that hypoglycemic event. Okay, so to think about the modeling choices we have here, let's assess our predictive performance in terms of just root mean squared error over the 30 minutes following exercise. And one model we can look at for forming these forecasts is a mechanistic model. So there's this popular UVA Padova simulator. It's FDA approved as a substitute for preclinical trials and the development of artificial pancreas algorithms. But it's a very rigid model and really struggles to model real CGM data well. So as an alternative, we can consider the host of black box sequence models. And not surprisingly, these have improved predictive performance. But again, that's only half the story. A question is, do they form reliable counterfactual inference? And what we see is that despite having good RMSE, black box models can violate known causal rankings of interventions. So for example, we know that the more carbs you eat, the higher your glucose will rise. Yet the black box models can predict exactly the opposite as happening. And a question is, how often do our models get these rankings wrong? And to study this, what we're going to look at is something we call causal error rate, where we define these intervention sets. So it's just a set of interventions where we have domain knowledge about which intervention has the largest effect. So for example, in this here, eating 100 grams of carbs would increase glucose most. So for each one of these interventions in this set, we form our counterfactual simulation under our trained model and use this to derive which intervention has the largest estimated effect. And so to form our causal error rate, we're simply looking at an average over all the different intervention sets where we have some domain knowledge about these rankings and saying how many times does the estimated max impact intervention not agree with our ground truth domain knowledge. Okay, so in our experiments, we're considering intervention sets of size 3, so random guessing would produce a causal error rate of 2/3. And what we see is the black box models are basically no better than random guessing. On the other hand, our mechanistic model has much lower causal error rate, having baked in all this mechanistic knowledge of insulin glucose dynamics. So what's going wrong with these black box models? Well, one thing is the fact that we're learning from just partial observations. There's this very, very complex physiological process with many unobserved sources of variation. Another challenge, though, is the fact that we're learning from observational data, right? So, for example, insulin dosing very commonly co-occurs with an upcoming meal. And so the model may incorrectly learn that every time I dose myself with insulin, glucose rises because I'm going to eat and that will bring my glucose down. Definitely not what's actually happening, but the model could learn this. And so a question, though, is whether we can rescue the causal validity of these black box models. And the answer is yes. With just a small tweak to the objective function, where we penalize for these misranked intervention sets that are in our bag of domain knowledge, we see a dramatic reduction in causal error rate. So in particular, we consider this hybrid objective just a convex combination of a standard predictive loss with our causal error, and this tuning parameter alpha tunes between our emphasis on predictive performance versus causal validity. And a note that for our gradient-based learning, we take the 0-1 loss that appeared in that causal error and replace it with standard cross-entropy loss. Okay, well, you might be sitting there, maybe Matt is sitting there saying, well, why do we care? He's not saying this because he knows about this, but he maybe before working on this would have asked the question of like, why do we care about this hybrid loss? Can't we just go back to our mechanistic model? We saw it had good causal validity. It wasn't flexible enough. Can we just make that model more flexible? So instead of a hybrid loss, what about a hybrid model? And indeed, this is the perspective that my group took for quite a while, spent a lot of time together with Matt working on various hybrid models. And so to see what happens, let's just start by assuming we have some mechanistic model. So modeling somebody's physiological state driven by some set of external inputs like consumed carbs and delivered insulin. And there are many ways to hybridize these models. One is to assume that the residuals are captured. You're going to model those residuals using a neural network. But there's a whole host of other approaches. So you can, if we walk along the spectrum, start with our pure mechanistic model. Another approach assumes that the simulator parameters of the mechanistic model vary flexibly with neural networks. You can also assume that the state interaction structure is maintained, but each state update is going to follow a neural ODE. Or you can forget the mechanistic model altogether and consider a black box ODE, neural ODE. Okay, but the question of interest here is as you walk along this spectrum, and the models are getting more and more flexible, is the causal validity that was encoded in this pure mechanistic model maintained? And when we add our hybrid model zoo to these comparisons, we see that we do get state-of-the-art predictive performance, leveraging the inductive bias from the mechanistic model. But as the models become more flexible, we have this increase in causal error. So hybrid modeling is important, but the loss is also important. But we can also train these models with a hybrid loss, and doing so really gives us the best of both worlds, where we have state-of-the-art predictive performance and negligible causal error, even lower than the pure mechanistic model. The take-home message here is that inductive bias is really key. So we have domain knowledge about some basics of insulin-glucose dynamics. We have some knowledge about treatment effects. We don't know exactly what the treatment effect is. We don't know it in all scenarios, but we do in some. We know the ranking in some. So leveraging this is really important even in the age of deep learning. So that's the take-home message here. And I'll just describe very quickly one other area where we've leveraged this type of hybrid modeling for unsupervised representation learning with the goal of subtyping patients. So patients have very highly varied responses to what they eat, and to better understand different interventions that we may want to give or disease progression, we'd like to be able to subtype patients in some low-dimensional embedding space. So we take our high-dimensional observations and we embed them in some low-dimensional space, and then we can think about clustering to define these subtypes or define axes of disease progression. But to make this space clinically actionable, we need these dimensions to align with clinical reasoning. And so what I'm showing here are embeddings of patient meal data for individuals with type 2 or prediabetes in terms of known correlates of disease, like insulin sensitivity. But insulin sensitivity is very challenging to measure directly, so how are we doing this? Well, to start, we're going to think about just taking a variational autoencoding perspective to embed our observations, but if we use a standard VAE, our dimensions are completely uninterpretable. And to see the lack of physiological semantics in this embedding space, we see that we haven't learned any features that seem to distinguish between these two populations. So instead, we can again leverage hybrid modeling where we take our mechanistic model and use it to ground the decoder which implicitly defines our embedding space in terms of these clinically meaningful quantities. And as we look across our embedding dimensions, what we see, again, these dimensions have interpretable meanings, and we see differentiators between these populations that align with things that are known in the literature. But the same is not true for the standard VAE. Okay, so stepping back, what we really see is the promise of leveraging causal domain knowledge, either through mechanistic knowledge or knowledge of treatment effects, but there's a whole host of other interesting questions that are still out there to pursue. Like, what if you only have partial mechanistic information? Or what if you have it organized in some multi-resolution scale, or through LLM agentic reasoning? And this is really just a push to encourage you to think about these ideas and leverage the inductive bias that you have, and really, rather than just turning to the latest, greatest black box model and focusing on predictive performance, hopefully in the two parts of this talk, I've hammered home the idea of how key it is to try and think in terms of causality and try your best to get at causality behind these different systems. And so to conclude this part, I just want to thank my amazing collaborators here, and in particular the students and postdocs who do the real heavy lifting on the projects. Shout out to Matt, who's in the audience here. Worked with him for some years, all the way going back to when I was at Apple. So thank you.
Thank you very much, Emily, for a really, really wonderful talk. So we do have some time for questions. So I think they'll walk around. I can also walk around. I hardly see you though. So, but maybe I can already get started. So in the POSH ALS data, how many of these hits already had genetic evidence?
Like existing in the literature? No.
Well, you also did the GWAS, I guess, right? So how many of your hits that you got?
No. So in ALS, we're actually just working with
POSH.
Yeah, we don't have the clinical data there.
Hello, thank you so much for this super interesting talk. I was wondering about the sparse autoencoder you showed. Something you mentioned was with that you pass on the embeddings that are more interpretable to domain experts to work with that. And I was wondering, what does that look like? Because to me, it's a vector, right? How can domain experts work with those embeddings?
So that's a great question. There's a number of parts to that. One is, okay, I'll start with the simplest answer, which is for each dimension that we think is meaningful. And then the interesting part is, what does meaningful mean? But for every dimension, you can do conditional image generation to provide visualizations of like what happens as you turn up or turn down the knob on activation of that feature. So those were the visualizations that I was showing in ALS. A question is, these are super high dimensional representations, so which are the features that you show? And the first pass is thinking about differential activation to understand which are relevant to, again, like healthy versus sick, perturbed versus control. But there's still a question of, there's still potentially a number of dimensions. And so we do various things there. Again, I can't go into the details of like exactly what we do, but we have ways to think of like what are the top few dimensions that we may want to look at. And there are different ways to think about reasoning over those. But yeah, when I'm talking about engaging with a domain expert, I'm thinking through the visualizations that have, again, tend to, not always, but have one thing varying. So versus like when you look at your dense SSL embeddings, the domain expert goes, oh, I don't know what it's trying to capture, right? Yes.
Yeah, it's a super exciting talk. I actually have a question for the HepG2 that you mentioned.
Yeah.
So like there's, it's really exciting to see like data set with like both modalities. I'm actually especially interested in the label-free one. Is that you have fluorescence, like the label-free, do you see it bringing more information that's unique to the label-free data, or is it like Regeneron recently announced that they think label-free can replace the cell painting panel they've been doing? I'm curious on your thoughts.
Wait, sorry. So is the question, how much do we believe in label-free?
How
Okay.
much do we?
So one thing to note in terms of like the Regeneron is they use Brightfield. We're using QPI and other versions. So there's an important difference there. We believe there's an important difference there. And then how much do we believe? I don't know that I can speak for the company in terms of making a statement right now on that. I think I can say we believe that there's significant information in label-free, and I think it has significant capabilities, including for things like live cell, and yeah.
Thank you.
Thanks, Emily,
Yeah.
for a great talk. Just thinking a little bit about the example with diabetes.
By the way, I feel like I'm back in my thesis defense. Windsor was on my committee. So I'm sweating.
Knowing what you know from your thesis, the question of causality, when you think about the observations as inputs and outputs, timing diagrams give you a lot of information about what causes what. And so as opposed to lumping it into a model and then getting confused about so how much of that are you distilling as an input-output model?
So are you talking about the second part I'm assuming?
The second part.
Yeah, so my group did a lot of work on Granger causality back in the day. So I have a lot to say about that. That's not really causality, right? But it is getting at your point of the timing information. And I mean, like these ODE-based mechanistic models are doing that through like modeling state dynamics and the interaction between states. There is a notion of this state leads to a change, standard, you taught me state-space models, some version of controls and long back in the day. But yeah, so there is that information there in terms of everything. So let me actually pull up one slide, which maybe, and Matt is absolutely, I'm sure, going to disagree with this statement. Feel free to do so. Where's the hybrid model zoo?
That's so exciting.
Wait, yeah. So this is actually my favorite version. This one and this one. Matt loves this one. I don't know. You can disagree. Do you agree? I don't know. Okay.
I agree that I love it. It's just, yeah, I don't love anything. No.
He hates his work. He hates his work. Okay. No, but.
I thought the right answer was exactly where you're going.
Okay, yeah. But both of these versions maintain the notion of one thing leads to the next and the interaction of them. Yeah.
Hi, thanks for a great talk. Just a quick question about these hybrid models and the hybrid losses. I guess it's two questions. So first off, generally when you're sort of in this, I guess, hybrid space between something that's purely like mechanistic and something that sort of has free range over your entire like model space,
Yeah.
do you see that giving this mechanistic understanding actually sort of generally improves the performance over the two extremes, or is the shape kind of more U-shaped?
So this is walking. Okay, so if I call this pure mechanistic, what is it called on the next slide? Hybrid sim is this one. This is M node, and this is B node, black box neural ODE. That's what's shown here. So this is mechanistic model, allowing the simulator parameters to change, allowing this is a combination with neural closure. Slight caveat in what I'm saying. This is maintaining the state interaction structure, but allowing each state update. So each linear ODE gets replaced with a neural ODE, but it's constrained in terms of what variables it takes in and which variables it affects. It's constrained according to the same state diagram in the mechanistic model. And this one has, the lowest RMSE. There's not quite, I guess, monosemanticity here, but monoticity. Okay, it's been a long day. Mono something. Okay, I can't speak at this point. So maybe that gets at what you're saying. It is not a hard and fast rule. It's a general trend.
So like to some extent, there is like this slight trade-off between sort of your original like RMSE objective and sort of your like
Yes.
causal validity.
Yes.
So I guess
I would definitely say this is, again, it's not formally, I've kind of organized as best I've experienced these things work. This is like reducing RMSE, but also increasing causal error rate.
So then I guess the follow-up question to that is particularly for the hybrid losses, where you
Yeah.
actually have a parameter that sort of dictates
Yeah.
how strongly you're enforcing your like constraint.
Yeah.
Do you guys have like a recipe of sort of how to choose that parameter? Like, is there some delta of like additional error that you're willing to accept, or is it sort of very domain dependent?
Yeah, it's a great question. I mean, you can treat it as any tuning parameter in a model, but I would say there's a lot of still room for thinking of it as like a domain.
Right. Yeah, like, cause I mean, normally in tuning, you're just like, okay, well, I'm going to do a grid and then say, okay, which one does the best, but it's not even clear here what the best is, because you have like.
Well, I mean, you have a loss, right? Like you have.
But you have kind of two losses that are sort of, to some extent, diametrically opposed.
But holding out data, like, like standard way of tuning these kind of parameters in models. But you're right that there is something still fundamentally different than like when you have an L1 penalty. Like you could think of, you have a tuning parameter if you do lasso, right? And so you think of that as a mix of fit with your L1 loss or something like this, but it is a little bit more nuanced here. I will definitely say that.
All right, thank you.
Yeah.
Thank you. So maybe we take some more questions just because it's time
Yeah,
that
absolutely.
they can just come directly. But thank you so much for a really wonderful
Thanks
talk.
everyone.